Welcome to the seminar of the Center for Genomic Science Innovation. So today it's my pleasure to introduce Kranthi Varala. Kranthi got his PhD at University of Illinois working on plant genomes and intra-species variation. And he continued as a postdoc at New York University, working with Gloria Curuzzi, where he used systems biology approaches to understand plant responses to nutrient supply, nitrogen in particular, with Gloria, and using also phylogenetic approaches to understand the genetic basis of plant adaptation to these environments. I need to say that it's during that time that I met Kranthi, and we got a really cool paper together, close genetics, using these techniques. And then later, Kranthi got an assistant professor position at Purdue University, and he started his lab again on plant responses to their environment and transcriptional regulation. So overall, he's using systems, as you're going to see, system biology, machine learning approaches to understand how transcription factors control the regulation of gene expression and plant phenotypes. And he's also very interested in evolutionary approaches, in particular, the conservation of these responses on these networks across different plants and that are covering broad phylogenetic distances. And Kranthi, over the last years, got really good funding from NSF, USDA, and published really great papers. So join me to welcome Kranthi to Madison.
Thank you for the introduction, and thank you for this opportunity to talk about my work. As my title says, I'm going to talk about network inference from public data sets. We've worked on this approach, and we have some really cool results from it. But before I tell you that story, I'm going to tell you a little story about why we felt the need to do this. So I'm interested in plant responses to the environment. So plants sense a lot of factors in their environment, the light, quality, and quantity of it, temperature, nutrient availability, salt levels, biotic interaction with other organisms, and across a broad set of plant species, they alter their growth and behavior to best suit that environment. That it's called plant plasticity, and that can be observed at levels of photosynthesis adjustments, the secondary metabolites they make, the homeostasis they maintain, the growth plasticity, the size and shape of the plants literally, and the developmental timing of that. At a molecular level, that happens by sensors that are typically embedded in the membranes, outer membranes of the plants, and they can sense all these environmental factors. And when there is a change in that environment, that triggers changes in the gene regulatory networks, which sense that change and respond to it, so that eventually the plant ends up in an altered transcriptional state that is better adapted to use those resources. So I'm going to focus mostly on this initial bit of sensors and gene regulatory networks. And just to give you a sense of the time scale of that, so the sensors that are typically embedded in the membrane, we don't know all of them, but we know some of them. They respond to changes in the environment within seconds, up to a minute or two, right? That's how quickly the plants know something's changed and start responding to it. And those typically trigger a set of transcription factors that are called the first wave transcription factors that are commonly post-translationally modified. So the protein's already there. It undergoes some kind of a modification that changes its behavior, either entry of the nucleus, forming a dimer, or some such interaction. And that happens within the first 10 to 30 minutes. And in that time frame, the first wave transcription factors are going to trigger other transcription factors that are downstream of it, in addition to some metabolic genes as well. And that's a time frame of between 15 minutes to about an hour to 2 hours. So this entire altering of the transcriptional state of the plant, which results in changes in metabolism, physiology, growth, and maturity, can happen pretty quickly within 1 to 2 hours of the change in that environment. And in terms of number of genes involved at each of these stages, we don't know all the sensors, so I don't necessarily want to speculate, but it's probably a handful of them. And for any given change in the environment, that first wave of transcription factors are typically 10 to 20 of them, sometimes less, right? And that set of first wave transcription factors typically set off between 200 to 300 transcription factors downstream of them, which are then going to go and regulate 2,000 to 3,000 genes. That's in a plant genome, that's roughly 10% of the genome, right? That's whose expression is altered in response to that change in the environment. So that's the kind of the scale of the problem. So in my lab, we use two approaches to try and understand how that gene regulatory network functions and how it responds. The first story I'm going to tell you is about trying to learn those gene regulatory networks in a very dynamic way by using time series data. The idea here is to use a very fine scale time series that's going to sample repeatedly in the framework of when that transition is happening. So that defines the universe of change. That's the set of genes and transcription factors that are responding to that specific stimulus. And from that universe, we try to infer the regulatory network operating within it. So this is a very carefully controlled experiment, and I'll show you one example of that. The other approach we take is to build this steady state gene regulatory network. So these are gene regulatory networks built from a very wide range of data, very little control on how those experiments are done. But because of the depth of information that's available there, we can build the relationships between which transcription factors are likely to regulate which genes. An important part of that is collecting the cohesive data set. And then an important innovation we did in this part, in this side, is to break that down into an organ level and then do a network inference. That will happen in the later part of the talk. So an example of that carefully controlled fine scale time series. So I work with plant tolerance to stress. The specific example I'm using here is cold stress. Tomato is a cold sensitive plant species. So when tomato plants are exposed to cold stress, they undergo heavy wilting. There's a lot of physiological changes. And part of the plant trying to compensate for that tolerance is the response that it generates. So when a plant is cold stressed, it tends to generate more soluble sugars to maintain the solute concentration. It generates a special class of proteins called cryoprotectants that protect it from the lower temperature. It generates membrane lipids to repair damage to the membrane, reactive oxygen species mitigation, and protein turnover. So we know all the processes that happen in response to the cold stress. That's the kind of the defined universe of what happens in the plant. At the same time, there are negative effects as well. It downregulates photosynthesis, it downregulates plant growth. What we don't know very well is what is the gene regulatory network that governs that response? How is the overall network altered in response to that stress? Which TFs are responsive, and which of those TFs govern each of these mechanisms? Like, is there one or small number of TFs that are responsible for generating cryoprotectants, for example, right? The challenge with determining this is that the GRN response that I'm looking at here happens within minutes, right? So how do you sample a system that's that dynamic? So first we had to come up with a way in which we could control the initiation of that stress to a specific time point. And our very high-tech solution is right here. It's a plastic tub wrapped in duct tape. And in that, we have a little 3D printed plate with 8 holes in it. Each of those holes is going to hold one tomato plant in a little foam plug. And the advantage of this system is that when I need to initiate my treatment, I can simply lift up this plate. So it has a full intact plant with the shoot system and the root system, and then move it to a different growth chamber that's already at 4 degrees Celsius and has pre-chilled water. So the entire plant shifts from 24 Celsius to 4 Celsius instantly, right? So once we have that, we can control the initiation of that stress. And we can also sample shoots and roots independently, so we can look at different organs. So we did that with plants, and then we sampled them at 15 time points in a 2-hour time frame. Initially 5 minutes apart, then 10 minutes apart, and then further down 15 minutes apart, right? So that's a pretty close sampling of plants. From that, we can identify genes that respond to that cold stress. We can learn the causal gene regulatory network, and then identify TF hubs from that. Just to show you an example of what the data looks like, all the samples in green here are the plants that stayed in ambient conditions. And there is a difference between them because of the diurnal regulation of genes. So all plant genes go through a diurnal cycle. So there's a little bit difference in gene expression over a 2-hour time frame. And that's what you're looking at here. The numbers in blue are the plants that are in cold stress. So you can see basically the trajectory of that altered transcriptional state. At 5 minutes, the plants pretty much look like the ambient plants. 10 minutes they're further apart, 15 minutes they're further apart. And then it slows down the rate of change. And down here is the last time points of sample, which is 105 and 120 minutes, right? So that's the speed of transition happening right there. So when we look at that set, that's about 7,000 genes that respond within that 2-hour time frame, which includes 500 transcription factors, which is a little bit disheartening when you're trying to infer a gene regulatory network, because now we have a problem with the thousands of genes and only 15 data points that we can learn from, right? But we kind of got lucky a little bit because when we started looking at the data, something we realized is that cold stress dampens the circadian oscillation in clock. If you don't know what that is, there's an internal circadian clock that operates in all organisms, and that alters the expression of genes throughout the day. And this particular gene PRR7 is one of the core components of that clock. And the orange line is what that gene is doing in ambient temperature. From the start of the experiment to the end of the experiment, that gene should go up. What's happening in cold stress is essentially flat lines, right? So that gene expression is stuck at wherever it was at the start of the treatment. And because the core clock is doing this, all the genes that it regulates are also doing the same thing. The things that should go up or go down in that time frame are also flatlining, right? So we call these genes cold static genes, and turns out that about 80% of the genes that we find cold responsive are in fact cold static, right? So what it's saying is the cold is stopping the clock and therefore stopping these genes from cycling. So from perspective of how is the plant responding to cold, these are not likely to be responsible for building that tolerance, right? So I can exclude that out of my inference analysis. And that reduces it to about 2,000 genes and 200 transcription factors. So then I can say, okay, these are the things I'm trying to model, which is a much more manageable set of genes. We did our TRN inference using an approach called dynamic GE3, which is a random forest-based approach built for time series data. And from that, we are able to infer gene regulatory networks. But before that, we can also do this. We can say, okay, here, if I just look at the trajectory of gene expression in cold stress, right? So this is the gene expression at time 0 for one of the genes, and that's its measurement at various time points. So for every gene, I can assign the time point at which that gene responded to cold by setting some threshold. So I can, here I'm saying 2-fold change, right? So the gene expression has to increase or decrease by 2-fold. And I can precisely point to the time when that happens. So that's my time assignment for that gene. Gene X responds to cold at 15 minutes, gene Y responds to cold at 30 minutes, right? So that gives me a linear time-based hierarchy. And after inferring the gene regulatory network, I can essentially create this, this flow. Each of these things, green triangles here, is a transcription factor. And each of the edge, say, is a prediction for that transcription factor regulating another transcription factor, right? The whole network is too large to visualize, so I'm just doing the transcription factors. And they're organized by time. So I have these 44 transcription factors that respond within 5 minutes to cold, and I have predictions for how they're going to regulate downstream transcription factors as well, right? So you could go in there, you can zoom in, pick one of those transcription factors and say, hey, what is this transcription factor doing? And you can pick out all the other TLs that's regulating at various time points. And you could zoom in further and ask for regulatory loops where you begin to see feedback inhibition of that original transcription factor. So there's an example. The same transcription factor within 5 minutes is inducing this transcription factor, which responds in 15 minutes. And at 40 minutes, there's a third transcription factor that's activated that goes back and inhibits the first one, right? So that's a very classic feedback loop. And this is great. We can find a set of transcription factors that are responding to that stress. We can determine the hierarchy in which they are doing so. And as I said at the beginning, we know the different processes that respond. But to assess which transcription factor is regulating which of those processes is harder, because the network inference did not give us enough confidence in those individual regulations. And the problem is fairly obvious. It's that we are trying to model a lot of genes with very few data points, right? And this gets frustrating. At this point, we've done a very careful experiment. We find all these cool genes, but if you had to pick individual things from that, that's when you slow down. You say, okay, I have to pick some transcription factors that I can validate. So I'm going to pick things based on some kind of heuristic for what's interesting to me. The heuristic we used is that we said, okay, we'll pick transcription factors that are responding early, have a strong response over time. So at the beginning, the normalized expression for this gene is somewhere in the hundreds. It goes up to about 15, 16,000. So that's a huge fold increase across time. And then we additionally required that it respond to cold in another species as well to look at conserved response. So we ended up picking 12 transcription factors that span this time course. And we generated CRISPR-Cas knockouts in tomato for each of them. This is just one of those examples. 2 guide RNAs per gene. You knock out the transcription factor. And then you can take those knockout lines, compare it to the wild type, and determine what are the genes that are regulated by that transcription factor. So I'm showing here an example of 5 of those. These 5 transcription factors are pretty early response. That's 15 minutes, 10 minutes, that's 20 minutes. And this is an upset R graph, which shows intersections between sets. So if you take this example here, these are the set of genes that are regulated by this particular transcription factor exclusively, as in they are not regulated by anything else. And that points us to this transcription factor controlling gene expression of genes that go to the plastid that's involved in photosynthesis. If you look at this set here, this transcription factor zinc finger 3 is going to regulate a set of genes that are involved in lipid metabolism and generating those cryoprotectants, right? So you could do this, you could learn each individual transcription factor, you could do overlaps, you could do combinations, but that's pretty time consuming. If you remember, there's hundreds of transcription factors here, and trying to determine each one of them this way gets frustrating, right? So in this project and the previous project, we kind of end up at the same spot. So what I tried to do is there a way in which we can build these gene regulatory networks from much larger data sets where we have a lot of samples, and that can give us some confidence about what are the genes and processes that are regulated by every transcription factor, right? The idea is if we can build that network across a lot of the data points, then we could take the set of genes that are predicted to be regulated by one TF and overlay that on my process. So how many of them are responding to cold, and then kind of estimate which transcription factor is regulating which process in that response, right? But to do that, you have to be able to build these genome-wide studies. So time series data, just to wrap up on this portion, we can do these genome-wide studies, especially in time series. It'll tell you the one or more transcription factors that are involved in that response. If you do a careful time series, you can get the hierarchy of that response. And typically you'll find 100 to 200 transcription factors in that. You can classify them early versus late, so you can assign relative importance in that response. But the GR inference is limited just because it's really hard for one experiment and one lab to generate enough samples to feed the inference algorithm, right? So ideally, we would like to know this broad regulon. So that's where the second part comes in, which is looking at how can we build these gene regulatory networks that tell me something about every transcription factor and every gene in the genome. So if we could build a genome-wide GRN, it would have kind of a general regulatory framework for all genes. And once we have that, if there's a particular trait or process I'm interested in, I can ask, what are the transcription factors that regulate this process, right? Or conversely, I could say, if I have a TF that I'm interested in, what are the genes that it's going to regulate, right? Those are the 2 kinds of questions you can answer. So to do a genome-wide GRN, let's consider the complexity of that. These are 100 seed plants. Highlighted in the middle here is Arabidopsis, which is a model plant that we've done a lot of experiments on. It has about 30,000 genes. Overall, the range of genes goes from about 20,000 to 50,000 roughly, with a few exceptions, right? If you consider the number of transcription factors across the same 100 seed plants, there's about 6% of the genome are transcription factors, and that's fairly constant across the genome. And Arabidopsis, which I'm going to talk about mostly today, has 1,800 transcription factors, right? So just as a theoretical possibility, right, there's anywhere between 20 to 200 million edges possible between a transcription factor and a target gene. That's a very large number. if we could get at it if we had thousands of observations, but as I said, any single lab or group would have difficulty generating that. But we do have a lot of public RNA-seq data, right? So when we started looking at this about in 2019, we estimated there were about 18,000 individual RNA-seq experiments in Arabidopsis. Now, they were done by anyone working on Arabidopsis across the world. There is no standard set of conditions. There's no life developmental time point or treatment common to them. It just represents the entire diversity of gene expression in Arabidopsis grown in all different conditions, right? So there's a lot of variation in there, but that's not a bad thing, because when you have this variation, you're actually better able to discern relationships between TF expression and the target expression. Okay. So we could take all the 18,000 data sets and try to build one network from across them. But my experience working with large transcriptomes from multiple organisms has shown me that, especially in plants, has shown me that the biggest delimiter for gene expression is organ. A leaf in a plant will look like a leaf no matter what condition you grow in, right? So it has a set of genes that are expressed there. It has a pattern of a global transcriptome expression, right? So if I try to build a gene regulatory network that goes across all the data sets, the information that I'm getting from one organ may be negated or at best not supported by the data from a different organ, right? So, and that's mostly because of availability. If you think of a transcription factor that can regulate 100 genes, not all 100 of those genes are going to be available in all the organs. In some organs, due to epigenetic modifications, those genes are closed and not available for regulation. And that's going to be this, the fact for that organ, right? So if organs are the main delimiters for gene expression, we really should be trying to learn these gene regulatory networks on an organ level. Okay. So to do that, we need organ level data sets, not 18,000 experiments, but in fact, a leaf data set, a root data set, and so on. To do that, we first looked at all these experiments have descriptions deposited with them. So we looked at the metadata, and as Colin will tell you, that's not a very useful thing necessarily, because lots of times there's missing metadata or not enough metadata to tell you what the organ was for each organ, right? So depending on those would be unreliable. So what we did instead is I asked pretty much everyone working in the lab at my time. That's three of them. Tina was a postdoc, Xiaojin's a grad student, and Madison is one of the undergrads who worked on this. So for every day, they would read a whole bunch of descriptions of this metadata. If there is good description for a sample, they would label it. If not, often those refer to a paper. They would go back to the paper and read the paper to see where the organ came from. So they did this for months, 2 or 3 months maybe, spend some time every day on it. And at the end, we were able to generate a labeled data set, which had, wherever possible, at least 1,000 examples of gene expression that came from one organ. Sometimes we didn't have 1,000 because organs weren't sampled enough, but we were able to build this. And with those labels in place, this is a reduced dimensionality plot, which shows that. So everything in green here is a leaf sample. Everything in blue here is a seedling. Then we have roots in brown and various fewer tissues here. There's a seed in there, a little black samples there, right? So what this shows is that at a global expression level, you can kind of cluster each organ by itself. Not by a very simple cluster, but you can see these groupings that exist within that overall expression data set. So how do we do this in an automated way? Because anytime you do this manually, there is a possibility of error. There's also a possibility of bias towards experiments that are better labeled. So we wanted to build a completely automated way of doing that. So we trained a classifier via machine learning. We used the labels that we knew and said, train from this, and then if I give you a gene expression set, all it has is a number saying this gene was expressed at this level, right? That's all I'm telling you. Then the classifier should tell me which organ it came from, right? Knowing nothing else about the experiment. So we built that classifier, and these are various metrics for assessing how well it worked, precision and recall and F1. are complementary. But what it shows here is that we have very high precision and very high recall, and therefore very high F1 for any of the organs that we had enough data to train with, right? So the leaf, the root, the seedling, when we have this number here shows how many samples we had in our in our overall data set, right? We have very high precision and recall. For organs where we didn't have a lot of data to train with, we have less reliable predictions. But overall, whether you count it as a macro or a weighted score, the precision and recall for this classifier was in the 80 to 90%, right? Which is extremely high, saying that we know nothing else about, the algorithm knows nothing else about the experiment except expression data, right? So Somaya and Sonali were two grad students who worked on this, developing this classifier. So with this, we had an organ-defined data set for each of these organs. So we can take that and say, okay, let's now try to infer a network for the root that only uses the root data set. Okay? And 766, as an example for root, is of course larger than any one lab would generate for one experiment, right? That's a large number of samples, so you have much more power. And there's nothing about Arabidopsis that's special here. You could do this in other species as well. As a test, we did a fewer number of samples in tomato. And in tomato as well, you can very easily train a model to recognize the organ from which a gene expression data set is coming from. And we've also done this with maize and rice and a soybean, and across all of them, it's fairly easy to build this classifier, right? Because the gene expression profiles are strongly indicating which organ is coming from. So we trained our model from this 4,000 samples in Arabidopsis. We used that to predict the labels of 11,000 samples, right? So that's our entire data set. Once we have each of those organs, we can then take that organ, and we used an algorithm called Gene3, which uses a random forest approach to predict which transcription factor regulates which gene, and it assigns a level of confidence to that prediction. So we can build the GRNs, and as a very uniform, unbiased threshold, we said, because the GRN is going to include predictions from every TF to every gene, a lot of them will be low confidence, so we're going to retain the top 10% of those based on that confidence value, right? So we get a leaf gene regulator network, a root one, a shoot one, and so on. So when you have these gene regulator networks, one of the ways you validate them is you look at specific predictions. So that's what that output looks like. This is a transcription factor, that's its target gene, and that's a weight, which is a sense of how well that's regulated. So you could take these table and say, okay, how many of these are already known or validated experimentally? And you could go down and count them. And you could pick samples like that from here. So for example, this transcription factor is MIB108 in Arabidopsis, and it's predicted to regulate a jasmonic acid transporter, and that's a pretty high level of confidence. And you could scan through literature, and there was a paper back in 2009 that showed that MIB108 regulates jasmonate-mediated stamen mediation. So in the flowers where the stamens are, MIB108 regulates the maturation of that stamen based on jasmonate levels, right? So that's a link that you could say that's validated, and you could go through and handpick these. But that's tricky because one, it's tedious to do, and second, there are only a handful of transcription factors for which we have known targets for that transcription factor at an organ level, right? So it's going to be biased towards what we know. So we tried to see if there's a way to better summarize this network. And one of the ways we came up with is to summarize each transcription factor as the sum of its weights, right? So we have the top 10% of network. So here's a little toy network that has 4 genes, A, B, C, and D. There's a transcription factor X and a transcription factor Y. X is predicted to regulate A and B, and those are the weights assigned to it. So the net weight of X would be just the sum of those two numbers. So that's 0.4, Y is 0.2, so X ranks higher than Y, right? It's a very simple system, and you can take each of these regulatory networks and produce a list of transcription factors that function in that network, right? So let's look at one of these examples. There's the top 10 ranked transcription factors from the flower network, okay? And highlighted there are the ones that are already known. So these have been characterized via genetic studies to play a role in flower development. So one is a classical example that controls petal development. If you knock it out, there are no petals. NAC2 controls the layer of cells around the ovule and ovule development, and that was already characterized. NAC100 controls the size of the petals, and so on. So each of these are predicted to regulate floral development, and we know that they have a phenotype. Now, one of the things that occurred to me when we're looking at these transcription factors is that, are we predicting a role for a transcription factor in an organ simply because that's where it is expressed? Like the simple explanation would be this transcription factor is expressed in the flower, therefore we could have predicted the role there. And that's true for some of them. So this particular DAS3 transcription factor does have a higher average expression in the flower, but it does have a little bit of expression other places. But SO1, which is the classic transcription factor that's characterized in floral development, is expressed in multiple organs at about the same level. But yet it plays a very specific role in flower development that it doesn't in other organs, right? So there's more to it than just where the TF is expressed. And if you look at that more broadly, we looked at the top 100 transcription factors in each of those organs. And if you look at this initial bit here, most of those transcription factors are predicted to play a role in a specific organ. There's a smaller number of transcription factors that are important regulators across organs, which doesn't match the expression pattern because this is all 1800 transcription factors. And there's some of them that are specifically expressed in one organ type, but most of them are expressed broadly across all organs. Right, so you couldn't predict the role of a transcription factor just based on where it's expressed. You need to build these networks to be able to do that. So that shows, at least at an organ level, you can say which transcription factors are playing the role. But if you're interested not just in organ development, but in a specific process that whatever is the trait of your interest. There was this paper middle of last year, 2023, that was published that looked at phosphate responsive genes, right? So they did a phosphate limitation and supply experiment, and they identified the top 50 phosphate responsive genes in seedlings, which means they included the shoot and the root together in one sample. So these are the genes that are most strongly responsive to phosphate. So if you take those 50 genes, and only 2 or 3 of them were transcription factors, if you take those genes and ask, what are the transcription factors that regulate these genes, right? We could ask, because it's a seedling and included both leaves and roots, we couldn't ask that question across both. So in the roots, when we look at that set of genes, which are here in the middle, and each of these triangles are predicted transcription factors that regulate them. So I'm showing the top 20 of them. 4 of them have already been shown to regulate phosphate responsive genes, and in fact have phenotypes that respond to the phosphate supply. So these control root growth in response to phosphate supply, right? All of these are from the root. If you ask the same question in the leaf, we have a set of transcription factors that have pretty strong predictions to regulate this gene, but none of them have been characterized. We don't know what these genes do in regulating these genes. And a big part of that is the world of phosphate response has been heavily focused on how the roots absorb phosphate and how they assimilate it, right? So a lot of the experiments have been done on the roots. There just haven't been enough experiments on the leaf level. But phosphate is assimilated in the leaf. So if we're looking for additional ways in which phosphate assimilation can be regulated, those are novel candidates to investigate because they may control how much phosphate the plant takes up and how it's utilized, right? So given just a set of genes that you're interested in, you could find regulators for it in different organs. And then another process that we looked at was lipid biosynthesis, specifically in the seed. And the reason we picked this one is that this has been studied for a very long time. It's a very important agronomic trait. We care a lot about how much oil is made in the seeds. So biochemically, it's very well characterized. We know about 300 genes, mostly enzymes that are involved in making these lipids, right? So it's a very well characterized gene set. We know some of the regulators of those genes, and we know that all happens in the seed. So if we take the seed gene regulatory network and use these known genes as a bait, right, to discover what are the transcription factors that regulate them. Then we get a network which looks like that, lots of genes in there. There's about 100 transcription factors in there. But again, you could rank them using the ranking algorithm. And again, highlighted in green here are the known regulators of this, right? So 20 years worth of research has identified multiple regulators. So there's very classic ones like Wrinkle1, LEC1, FUSE3, which are known to alter seed lipid content. So you could expand that role to the 20 transcription factors. You get more known ones, but you also get many novel predictions of these are the things that should also be regulating seed lipid content. And just to see, does our approach work any better than any other previously built gene regulatory network approaches, which was a question asked by the reviewer. So we looked at that, and we can show that our approach of building these gene regulatory networks in an organ-specific way and ranking them based on that weight does better at finding the known ones, right? So these are precision and recall, which say how many of the known transcription factors did we find, and of the ones that we predict, how many are accurate, right? That's what precision and recall does. So we use the same query set of 343 lipid genes across these three algorithms, and ODGRN, which is in green here, has a higher precision and higher recall for identifying known regulators. Okay. So we found 8 of the 24 known ones. RNet found 6, and IGRN 2. And I should say that the most likely difference here is that RNet 2, which also works from all public RNA-seq data, does so without splitting it up into organs. So it's trying to learn it from all data put together, and there's probably missing some because of the internal conflict within that data set. IGRN2 does use organ level, but they included fewer samples just to reduce their computational complexity. So it probably just missed out on having enough information to do this. Okay, so both splitting it into organs and having enough data are both important for finding more. And if we had more data, we would probably do even better at identifying these. Okay, so just to give you an overview of what these transcription factors do, on the left here, so in the middle here are some of the genes that we know are involved in lipid biosynthesis. And there are different phases to this, like any metabolic pathway. There's the fatty acid synthesis, elongation, storage of those lipids, and all of that such, other offshoots of that pathway. And our network says these are the known regulators, and these are the points in that known pathways which it regulates. We also have these proposed regulators that are have predictions on what they regulate in here, right? So our naive algorithm, knowing nothing about seed lipid metabolism or about transcription factor regulation, just from gene expression is able to predict this. And then the question is, okay, you've proposed 12 novel regulators, how many of them are actually functional, right? So to validate that, what we looked at is the very common phenotype you assay in these kind of studies is you take the transcription factor, you mutate it, you grow them side by side with the wild type plants, and you check at the end of the life how much oil content is there in the seed. So if something that has a very large effect, you'll see a very big difference in the amount of fatty acids per seed. This is for 6 different of those validated transcription factors. In each case, you should see that, right? So to assay our knockouts, we were able to isolate homozygous mutant lines for 10 of the 12 candidate years. And for each of them, we grew 8 to 10 plants from each time, along with the wild type. And at the end of the life, we get 10 milligrams of seed from each plant, and we estimate how much is the total fatty acid content in them, right? And out of the 10 that we tried, 5 of the transcription factors have a large change, have a significant change, not necessarily a large change, in the total fatty acid content, right? So we are able to validate at essentially a rate of about 65%. So let me just focus a little bit as far as mechanism goes on one of these. So the one that had the biggest change was this transcription factor called MIBS2. So we took that one, we identified an additional mutant line, independent mutant line in that, and both the mutant lines showed a significant change in the total seed lipid content. In both cases, approximately 18% of the seed lipid content is reduced in the mutant line. We generated overexpression lines for the same transcription factor, and across multiple overexpression lines, you get approximately 12% increase in seed lipid content, right? So when you mutate the gene, you lose seed lipids. When you upregulate the gene, you get more lipids. So then to further characterize exactly how that's happening, we picked one of these overexpression lines. We did an RNA-seq of that overexpression line versus the wild type. And what we found was, I'm showing two of them here, but multiple members of this family of enzymes called purple acid phosphatase, PAP, are all upregulated when you overexpress this gene. And there was a paper last year that showed that these enzymes are involved in fatty acid biosynthesis in the plastids. So what we think is happening is that when you overexpress this transcription factor, it is increasing the level of expression of this class of enzymes, which increases how many fatty acids are being generated in the plastids. And because that increases, that then triggers the downstream increase in expression of multiple genes that are involved in turning those fatty acids into lipids that are stored in the seed. And we were able to show that in the same overexpression line, that all these enzymes involved in different stages of the lipid generation are all upregulated when you overexpress this transcription factor. Okay. So at least for one transcription factor, we've been able to characterize this. But zooming out, if we look at the overall profile, if we take, okay, here are the 20 transcription factors that are predicted to regulate seed lipid content, right? How many of them are accurate, right? So we have a 65% accuracy, which is pretty high for an algorithm that doesn't use any previous biological insights, right? We didn't tell it what each transcription factor is supposed to do. We didn't tell it what we already know in terms of TF activity. All it has is gene expression data. And from that, it's able to identify at about 65% accuracy. And so if that approach works, then that gives us confidence that we can then use these networks to we can query them with any bait, right? Any coherent set of genes. That could be the set of 100 or 200 genes that are most highly changed in the experiment that you are doing, which may have nothing to do with any of this, but whatever trait you're looking at, if you have a set of genes that you know are involved in it, either by an expression experiment, by metabolic pathway analysis, then you could take that and use it as a bait for this network. You could query the network with that bait and build your own little set of predictions for transcription factors that regulate that set of genes. And then you could prioritize those for identifying the phenotype that you're trying to alter, right? So now instead of having to work with all the transcription factors that respond, you'd be working with a subset of them. So it's a faster way to get to what you want to change in the plant eventually. Along the way, we've built this ML classifier that we think should be readily transferable to any other species. One of the things that we tried that I haven't talked about here is you could build your classifier in Arabidopsis, and you could use that to predict the organs in a completely different plant. The accuracy suffers a little bit. So when we tried building our model in Arabidopsis and try to predict the organ from gene expression in maize or soybean or rice, we get to about 65% accuracy, 65-70% accuracy, instead of the 80 to 90% we had in Arabidopsis. But when something is labeled as being from an organ, it's very likely to be right. So what you're missing out really is some of the things that don't quite fit that profile. So your recall may not be perfect, but the organ set that you're building in that species is going to be fairly accurate, right? And then that becomes the basis for that coherent gene expression set that you can then build your network from. Okay. So we are hoping to do this in at least the major organs, leaf, root, and maybe the seed. For multiple species, we already have the gene expression data sets for the major crops. Last time I checked, there's about 10 plant species that have at least 10,000 RNA-seq experiments in them. Some of them have much more. Maize has like 50,000 now. So that gives us a lot more data to work with, and we should be able to infer these networks from all of them. There's also been improvements in the algorithm side. So Genie3 is pretty computationally expensive to do, but the GRN boost is essentially using the XGBoost framework to improve the efficiency of Genie3. And we're hoping we can use those improvements to build larger data sets and use them for better networks. Our paper describing all of this work is on bioRxiv. It's also under review. We're hoping to hear back fairly soon from our second review. But we've also built a web interface where you could go in and use it right now, right? So I'll just wait for the video to start over here. At the very top, you have an organ that you select, and then you paste in your list of genes that you're interested in, and just hit search, and that should, within a couple of seconds, show you the top 10 regulatory genes for that particular set of genes. And there's a little visualization for that network, and you can get the full results of the entire subnetwork for all those TFs and all those genes from that page. You can download it and process it in any way you want. So we're hoping this will be broadly useful to folks beyond what we're just doing. And when we expand it to other species, beyond Arabidopsis, it should come in fairly useful for species that don't have as many genetic resources, where you can't just order up a mutant and try 20 different transcription factors. It would help greatly if we could focus on the ones that are most likely to give us a phenotype. I should acknowledge all the people who worked on this. We've had a great team in my lab who worked on developing that classifier and the GRN inference, a set of grad students and all the undergrads and postdocs who worked on generating those labels and developing all those algorithms and computational optimization to generate the network. A postdoc, Rajiv Ranjan, who's done most of the seed lipid metabolism work, and my collaborators, Ying Li and Karen Hertzen, both at Purdue, who've helped with the more technical molecular aspects of this, and my funding from DOE for this project. With that, I will take any questions. Yep.
I was wondering, like, to perform something like this, what could be the number of RNA-seq data?
What is the minimum number of RNA-seq data? So I talked mostly about the seed network at the end, right? So if you look at the number of samples, there's only about 100 samples from the seed. So it's not a huge number that are required for network inference necessarily, but that's depends on what's happening in that organ. So the seed is relatively simple biologically, at the risk of offending people who work on seeds. So seeds kind of accumulate a lot of lipids and protein and starch, and they store them, and they kind of wind down into a kind of a dormant state. So there's not a lot of different kinds of biochemistry that's happening in there, right? So that may have helped us in terms of inferring that network. While if you're looking at organ that's much more responsive, like leaves tend to do a lot of different things, photosynthesis primarily, but you have a lot of secondary metabolism and all that going on. So it depends kind of on the complexity of that, right? So you can only learn something if those transcription factors and then those genes are have a variation across the samples that you're looking at, right? So if you're studying some specialized metabolism, for example, that only happens in certain situations, then you would need a lot more samples to be able to see enough of those instances to learn that. Yes. Mm-hmm.
So you kind of get injury.
Mm-hmm.
Is that based on the knockout data, or were you purely just based on
those were specific predictions. The feedback loop that I showed here. Right. So these are network predictions, right? So we used a time series optimized network inference algorithm called dynamic Genie3. So it predicts what is the influence of one transcription factor or another. Then you threshold that, and so these edges are computationally predicted. To validate them, you would have to make the knockouts.
But is that prediction?
Yes. Yes. Yeah. So it's all it knows is essentially curves like this, right, for each of those genes, and it's predicting based on that.
Different plant sets temperatures, or I guess have a different threshold in this sensitivity. And some you wanted to have a clear shift from 4.
Right.
But I expect so many things digital the physical change actually happens. So even the sensor, the initial sensing process of that. So then the farther down the network much quite complex. So if you were to in the future do a shift at the more gradual manners, we expect to see a simplified responses and therefore tease apart some of the networks. And as a result, they infer what responses lead to the death of the patient or certain applications.
Yeah. So the temperature shift was dramatic to trigger a dramatic response, right? That was our goal here. A gradual shift in temperature, the problem becomes that for that species, you would need to know what the threshold is where it starts responding. Not knowing it ahead of time, if we knew it, like if we knew that 10 Celsius would trigger a stress response, then that's what we would do, right? We were fairly certain that this would trigger a response. But one of the things I mentioned, that cold stress stops the clock that we observed in our data. So my new grad student is looking at exactly that if you didn't shift dramatically the temperature, if you shifted from 24 to 10, for example, or another temperature, would you still get that? Is that an acute response to the sudden shift, or is it that something that's built into cold stress? So he's looking at that right now. And we're also doing that as a comparison between our Arabidopsis and tomato to say, do this, when we talk about temperature, the physiological effect on different plant species is going to be different. So how do we normalize that? Should we be treating all tomatoes at 10 Celsius and all the Arabidopsis at 4 to get the same response, right? So we're trying to, just using that clock stoppage as a clear indication, we're trying to do those experiments to see which is the optimal temperature. And the answer may be very different for each plant species. Yeah.
So in your GRNs, you predict your regulators and which genes are going to be regulated by these particular TFs. And so you also looked out some of these TFs. Did you try, for instance, to validate these predictions of, let's say, take a mutant in one of the TF and check that the predicted targets in that mutant are not regulated?
Right. So we've done things like that in the past, right? So BZIP1 was one of the papers we published from the Kranthi lab. And the answer is always mixed. You always get some of your predictions are right, that's the things they're regulating, and sometimes you don't, right? The problem, especially with not so much this cold study, but what the broad GRN is that we don't often know where that prediction is coming from. Like, we don't know which conditions triggered that particular response from that gene and was tied to that transcription factor, because we didn't control the experiments that were done, right? It's a very large set. So I suspect if we investigated it from that perspective and said, I'm going to try to validate each of these predictions by mutant studies, it would be a toss-up. We would get, for some transcription factors, you would get 20% of them validated. For others, you get 40% of them validated. You're almost never going to get 90, 100%, right? Just because it's very dependent on other factors that are working at the same time.
Yeah. They always question that with the increasing number of single cell data set, how do you think your analysis?
I think it would, in some sense, it would be better, in some sense, it would be worse. So when we take an organ and we are asking which transcription factor is regulating which gene, we are asking what is the mean expression across a population of cells. That isn't necessarily all doing the same thing, especially the more complexity to that organ, the more cell types in there, the more we're averaging, right? Once you get to single cells, if you get multiple cells from the same cell type and ask the same question, okay, I'm going to do a network inference in here, I think you'll get better clarity and better delineation of which transcription factors regulating which gene, with the caveat that single cells don't measure every gene, right? You get a readout of maybe 7 to 10,000 genes at the current technology level. Maybe that'll get better. So there'll be a lot of missing data, right? So as long as the inference can account for that missing data, like absence is not absence of regulation, right? We need to code that in some way. Then we can say, okay, then we can learn better from them. I think what will happen is we'll have to wait for enough single cell data to accumulate, and then again do the same thing where we take multiple single cell experiments, identify cell types from them in an unbiased way, and then from that unbiased set, try to predict our TF target interactions. I think we're not there yet, but hopefully we'll be there in a few years. I thought I saw another. Okay.
All right, let's thank Kranthi for a great talk.